Hello and welcome to the last day of MD and DI's Focus on Fundamentals course, Medical Device and Pharmaceutical Testing, Regulatory Updates, Trends, and Anticipated Changes, including FDA, ISO, USP, and MDRs, sponsored by Nelson Laboratories. I'm Chris Keach and I'll be your moderator today. We have just a few brief announcements before we begin. First, this webinar is designed to be interactive. The dock of widgets at the bottom of your screen will allow you to learn about today's speakers, download resources, share this webinar via social media outlets, and participate in our Q&A session that takes place at the end of our presentation. Please note that during our Q&A session, we're going to try to get to as many questions as we possibly can, but if we're not able to get to your question today, someone will get back to you via email after the session is over. And the slides will also advance automatically throughout the event. You may also download a copy of the slides by clicking on the Resources widget at the bottom of your screen. And towards the end of the webinar, we will ask you to complete our survey found on the right-hand side of your screen. Please take a moment to fill this out before leaving us today, as your feedback will provide us with valuable information on how we can improve future events. And lastly, if you're experiencing any technical problems, please click on the Help widget found at the bottom of your screen, or type your issue into the Q&A text area, and we will be glad to offer one-on-one assistance. And now on to today's class. Cleanliness testing for medical devices, updates, and trends. Discussing today's topic from Nelson Laboratories is Alexa Tatarian, Chemistry Study Director, Alpa Patel, Senior Scientist, and Paul Litley, Consulting Manager. To learn more about our speakers, please visit the Bios widget. And it's with great pleasure I now turn this special session over to Alexa to begin. Alexa?
Chris, thank you so much for that wonderful introduction. Welcome everyone to the webinar today. My name is Alexa Tatarian, and as Chris mentioned, I'm a Chemistry Study Director here at Nelson Labs. And today we're going to be talking about cleanliness testing. And if you've tuned in with us for the previous 2 days of this 3-day webinar, we spent a day discussing biocompatibility testing. We also spent day 2 discussing sterilization testing. And today we're going to be talking about the cleanliness testing of different medical devices and products. And we wanted to do this in its own separate day because cleanliness testing is really its own individual assessment in addition to biocompatibility testing and sterilization testing. And so we felt it was important enough because it really is its own topic that requires a different set of testing, and that's what we're going to be talking about today. So I'm going to kick us off today. And the cleanliness testing that I'm going to be talking about is really related for newly manufactured medical devices. We're going to be talking about residuals and potential contamination that's left over from the initial manufacturing and cleaning of devices. And so these kind of residues that we're talking about and potential contamination are those such as oils, greases, lubricants, detergents, polishing compounds, particulates, really from that initial manufacturing and cleaning of a device or product. And so those are what we are going to be first talking about today. I deal a lot with single-use devices such as orthopedic implants or spinal implants, a lot of implants, but we can also use this testing for reprocessed or reuse devices if the manufacturer is again looking to target those types of manufacturing residuals, so maybe first-time use, and then we'll discuss later today those reprocessing and reuse cleaning validations. But right now, starting off cleanliness testing for newly manufactured devices. So I want to first start off by mentioning this new ISO document. This is exciting. It's a new standard being developed called Implants for Surgery, Cleanliness of Orthopedic Implants. So it does have that word orthopedic in the title, orthopedic implants, but I will say that this can be used for really any device or product where you want to target those initial manufacturing or cleaning residues, but especially the bread and butter for this standard will be orthopedic implants. This is exciting and significant because it's the first time that ISO has gotten on board to develop a standard and guidance for this type of testing. We have had guidance currently from ASTM ASTM, which has a couple of different cleanliness standards out there, and there are some international standards as well that U.S. manufacturers and international have been following. And so this is exciting because it's the first time that ISO has really, gotten on board and felt that they needed to write a standard about this. And so to give everyone an update of where that standard is in the process, a committee on the ISO developed a document, and that went out for vote. It went out for an initial vote, and then people were allowed to comment and vote on that document. And that committee, which I have participated in along with some of my other colleagues here at Nelson Labs, and this committee had representation from U.S. as well as international countries. It's actually been a fabulous collaboration between many different labs and various experts in the industry on this topic. And we went through all the comments from that initial vote, and we revised the document. And through that revision process, we ended up making pretty significant changes and major changes to the document where we actually had to send it out for a second vote. And that happened a couple months ago where the document went out for a second vote. And so right now we're waiting to hear feedback from that second ballot, and we will start revising and reviewing those comments again. And so stay tuned for the official release of this document, but that's where we are in the process. Again, really exciting and significant because this is the first time that ISO has really become a major player in this cleanliness testing for orthopedic kind of single-use, newly manufactured cleaning validations. Just to kind of run through, now this is based on the draft document, so there could be changes here, but this is the general gist of the standard, what it's going to be about. The standard talks about how these cleaning validations should really take a risk-based approach in setting up the validation and testing plan. What that means is individual manufacturers will take a look at their process, their device, what comes in contact with the device, what could be left behind, this kind of risk-based approach of what theoretically could be there and what the manufacturers are concerned with in regard to the cleanliness of the device. And then that will help us choose different test methods to try and evaluate that cleanliness. And so the standard walks through that process of risk-based approach and how to document from the manufacturer's perspective. And the different types of tests that we're talking about, we look at microbiological contamination and bacterial endotoxin being potential target residuals, as well as organic residuals, and this could be hydrocarbons or organic carbon, and this also breaks down into both water-soluble and non-water-soluble contaminants on the organic side. And it also recommends to look for inorganic contamination as well. And then the standard also advises cytotoxicity testing. And so if you've been involved in this type of cleanliness testing before, none of this should be new or out of the ordinary for anyone. And so this document really isn't changing anything to the way that cleaning validations have been done ever since I've been on board for probably the past 10 or 20 years. So this isn't anything new, but it's ISO formalizing how all of this is done in an actual document. And then the standard also gives guidance on continued monitoring. I think manufacturers do a good job of setting up an initial validation, but often forget to set up kind of a routine monitoring plan so we can have a check on that cleanliness throughout the production of these devices and products. And so routine monitoring also takes a role in this new ISO document. I also want to transition really quick and give an update on what's going on with additive manufacturing or 3D printing for these medical devices and different test configurations. So recently, the use of different additive manufacturing processes and 3D printing has been increasing. We've seen an increase in the medical device and pharmaceutical space that more products are using this technology. And what we have found is that traditional test methods might not be appropriate to test these kind of devices. At the last ASTM committee, that was back in May of this year, I'm on that committee, and the Cleanliness Task Group had the FDA there, and the FDA presented on research that they've been doing on various additive manufacturing procedures. And specifically, if particulates are a concern for your additive manufacturing or 3D printed devices, routine methods to test cleanliness, specifically ultrasonic extraction methods, seem to be inappropriate to look for these particulates. So what's happening is some of these types of products have a lot of particulate residuals left behind after that additive manufacturing process, certain additive manufacturing processes. And what's happening is those particulates are getting caught in the texture and the geometries of the device. And you have ultrasonic extraction methods. Ultrasonic, for those unfamiliar, it's using sound wave technology to remove these residues from the device. You can think of it as more of a vigorous shaking. And those particulates will kind of, come off the surface a little bit and then just kind of float back down after the sonication process is completed. And so at this point, the FDA, other labs that are around, are investigating alternative test methods, or what is the best test method to look for these types of residuals, because ultrasonic is used in many different test methods to look for surface residual cleanliness, which is what we're talking about, and that is looking like it's not appropriate for certain additive manufacturing and 3D printed devices. So this has kind of got a lot of buzz in the industry right now because this is becoming a more popular manufacturing method for in the medical device space. So I would say if you're a manufacturer that does work with this type of manufacturing, just stay tuned and just know that if you have to assess cleanliness, that we might have to, you might have to work with the lab you're working with to develop the best test plan that's most appropriate for your device. And that concludes my portion of the webinar. I'll be back for the live Q&A a little bit later, but now I'm going to turn the time over to my colleague Alpa to talk about reusable device testing.
Good afternoon, everybody. I'm Alpa, and I will be giving you all an update on what is going on in the reusable world regarding medical devices and the standards that are applicable or documents that you can use to establish what you need to do or how to move on with how the industry is moving on with the reusable medical device testing. The European Union Medical Device Regulation, which was approved by the European Parliament, published in the Official Journal of European Union, is now available for everyone to read. If you guys haven't had a chance to read, I'll tell you it is worth reading that document. The new regulation is complex and quite lengthy, and the changes that it embodies are significant. One of the things that caught my eye is that all products have to be CE marked under the new regulation in order to be placed in the market or put into service. There is a lot of information regarding that in the MDR Article 5 if you have access to the document. They also give you a transition phase that if you can't meet by 2020, you have till 2024 to focus on how to get your devices CE marked for Europe regulations. Some of the changes in the MDR that should be noted is that some of the devices were reclassified. These were mainly in the orthopedic space regarding long-term devices like joint replacements and active implantable devices. These were classified from Class 2 to Class 3. There's a lot of information regarding the classification in the annexes. Rule 8 goes in great detail. However, I can't, if I had 2 days to go over this, I probably would, but in the time, I'm just directing you guys to where you can find more information regarding all of this. There's also changes in classification regarding active therapeutic devices. Closed-loop systems or automated external defibrillators will be classified as Class 3 as well. The Global Harmonization Task Force and Medical Device Regulators Forum pushed for unique device identification for global regulatory harmonization and have made this requirement mandatory if you want to market your device in Europe. The MDR requires the traceability of all devices placed on the EU market, except custom-made devices, by means of this unique device identification system. And the reason for them to push this this UDI system is to ensure that they can monitor the device through UDIMED, and it gives a database for the device. Some of the requirements include that each device will need to have an assigned UDI obtained from a UDI supplier, and this information must be loaded into the European database of medical devices. The UDI must be established before the product is placed onto the market, and the UDI carrier must be provided on the device, label packaging, and in case of reusable medical devices, in itself. And this document does go over that if you do have this UDI placed on a reusable medical devices, you have to do something called repeated testing to ensure that during the lifetime of the use of the devices, it does not fade away or end the numbers, or they are readable. With regards to UDIMED, all devices will have to be uploaded during its lifetime of use. Class 1 medical devices should be uploaded periodically and should update post-market surveillance. Periodic safety update report for Class 2A, Class 2B, and Class 3 must be uploaded as well. Reprocessing of single-use devices is only allowed under specific conditions. Permission by the member state is one of them. And if you see Article 19 in this in the MDR, you will see that there is a lot of information regarding single-use reprocessing that Paul will probably go over and outline some of the specifics. One of the interesting things regarding single-use reprocessing is that the original manufacturer will no longer be mentioned on the label, but will be mentioned in the IFU. As for ASTM updates, ASTM is one of the most active committees regarding reusable medical devices. The committee that represents the medical and surgical materials is the FO4 meetings. If you haven't come to these meetings, we strongly encourage anybody who is involved in reusable medical devices to start participating in these meetings. As you can see, I have listed at least 3 working documents and one that we finalized this year. And there's still a lot more to come regarding reusable medical devices. There is a lot of uncertainty out there regarding how to do the validations and how to do the extractions, and what are the criteria for reusable medical devices and acceptance criteria. So ASTM is looking into every phase of a cleaning validation, breaking them apart and making them into working groups. And so each working group is focused on one phase of these validations and are focused. The good thing about this is that there is an FDA representative in each working group that participates as well, not only there at the meetings, but they're also writing these documents with us. Another one that is also up in revision is AAMI TIR 12. This is a compendium of process materials, test methods, and acceptance criteria for cleaning reusable medical device. And this is currently under revision, and a lot of work has gone into the revision. Some of the things that are coming out of the standard is that there is a proposal to add an annex which will help define cleaning protocols for reusable medical devices. What these protocols will entail is they will help eliminate all these different IFUs that are floating out in the industry and to harmonize them for one particular type of device. So far, a preliminary list has been identified where these devices have been categorized, and they have been 3 categories for reprocessing instructions. However, there is still a lot of work that needs to be done. We started off with 9, then moved to 7, and now we've come down to 3. But this is still very preliminary. But this spurred up when one of the manufacturers said that we have hemostats. And if you look at an IFU for a hemostat, you will probably see 20 different IFUs. How can a healthcare facility use that different IFUs for cleaning the same product? And that's what spurred up this annex, and that's what we're working towards addressing in this standard. As for Tier 30, which is test methods and acceptance criteria for cleaning reusable medical devices, is also under revision, and also a lot of work has been going into it. Some of the proposed items that will be discussed in the next upcoming meeting in October will be regarding TOC acceptance criteria, understanding benchmarks related to cleaning validations. Are we using the right benchmarks? Protein, hemoglobin, carbohydrates, TOC. What is the appropriate marker to use for testing? And language regarding meaning of simulated use. There is a lot of uncertainty out there regarding what is simulated use. How are we supposed to perform these tests? What does it entail? How many cycles do I have to perform? Nothing is outlined in any standards at this point regarding that, but we are performing cleaning validations with simulated use testing. So this was a method for us to bring it into this standard and give guidance to the manufacturers to what the validation would contain. Tier 30 will also be revised to be in line with the ISO documents that we have. The ISO 17664, 1583, which are for washer disinfectors, and the 2015 FDA guidance documents. And all of these topics that are outlined here are going to be harmonized somehow. We're going to make it so all the standards say the same wording. The ST91, this is for flexible and semi-rigid endoscope processing in healthcare facilities. This topic has gained a lot of attention due to scrutiny the endoscope received from the regulatory bodies regarding reprocessing in the last few years. This document is currently under revision, and a lot of work has been done in the documents to update the sections that needed attention. Some of the sections that were discussed in detail in this year's meeting was discussion regarding critical devices versus semi-critical devices for endoscopes and which category they belong to. However, because this is such a topic that that is really hard to put into a box, there was no resolution made. But due to Spalding classification, definition of critical devices, where these devices have to be sterile, is somewhat not a possibility for endoscopes, and the healthcare facilities at this point are not ready to take that on. So it's still an unanswered question, but we're hoping that in October we should be able to come to a resolution, or come at least close to a resolution to where these devices should be classified against. There was also discussion regarding surveillance of endoscopes. Because there's a lot of uncertainty regarding extraction and culturing methods out there, and also the healthcare facilities are not quite ready to do this at this time, this question is also unanswered, and it's still open to the possibility that it may be added or may not be added in the document. Discussion regarding training and competency of HCS staff, Human factor considerations, I'm sure you heard that word a lot in reusables. Because we had a lot of discussions that were unanswered, we had a stakeholders meeting on the 11th of September in Virginia regarding these topics. 3 presentations were presented by 3 professionals who are known in the industry. I'm sure you guys all have heard of them. Dr. William Rutella, Dr. Michelle Alpha, and Kari Ofsted. They've done a lot of work in the endoscope industry and are continuing to do so. All of their presentations were around discussion moving toward terminal sterilization instead of high-level disinfection of endoscopes, modifying and clarifying the Spalding classification, saying that these belong in the critical dimension and not as a semi-critical device. Studies showed that additional reprocessing did not help. Ethylene oxide sterilization is not the answer all the time. Liquid chemical sterilization is difficult to perform in healthcare facilities and monitor. And performing high-level disinfection twice hasn't shown any benefit either. They have proposed to the industry and to the standard committee to promote a new low-temperature sterilization process for these type of devices, as they believe that they should be sterile and not high-level disinfected. Another standard that's under current revision are the ISO 15883. These are the washer disinfector documents. The standards that are currently in revision are the ISO 15883-1, which is general requirement terms and definitions. I mean, it says it all just by looking at that. 15883-4, requirements and tests for washer disinfecting, deploying chemical disinfection for thermal level endoscopes. That is finalizing. However, I do believe that once we have cleared ST91 and come up with some resolution of how we're going to address this, it will still be open and we'll have more information to put into that standard. As for the -5 that is focused on test soils, that it's quite completed, but there is still a lot of debate about determining updated acceptance criteria for protein and hemoglobin as action and alert limits. They are still not sure if the benchmark specified in TR 30 is appropriate for Europe. Are they comfortable with those acceptance criteria? The ISO 17664 is also under current revision. This is about this standard is on sterilization of medical devices, and the scope was widened to include devices for which a terminal process is disinfection. So adding disinfection for endoscopes. NXA provides greater detail about process commonly employed by healthcare facilities. NXC was added to provide information on the classification of medical devices, and more guidance is presented in each section, and sections are broken out to avoid confusion. For example, the cleaning has 2 sections. Now it has a manual and an automated section. They have great details on what those contain. This ISO was also voted, so it will become part of an ISO document and replace ST81. What I'm excited about is the ISO 17664, medical devices not intended for direct patient contact. This is something that we're seeing very commonly being tested, and there's no guidance around it. So a lot of the times we're putting into scrutiny in as the disinfection acceptance criteria, and they probably don't fit in those Spalding classification either. This is a new standard, and it will start in December of 2017, which we hope to see a lot of work being done on that. Now I will be passing the phone to Paul Littley to talk about reprocessing of single-use devices. Thank you.
Thank you, Alpa. In the final section of today's presentation, we will discuss industry updates and trends for the reprocessing of single-use medical devices. For clarification purposes, a reprocessed single-use device, by definition, commonly refers to an original device that has been used on a patient and then subjected to an additional processing and manufacturing, primarily cleaning and functionality testing processes of the device for the purpose of an additional single use on a patient. Reprocessing of single-use devices is typically performed by third-party firms. For the remainder of this presentation, please note that for convenience sake, if I use the word reprocessing, I am referring to the reprocessing of single-use devices by third-party entities. Current consensus estimates that approximately 20 to 30% of U.S. hospitals reported that they reuse at least one type of single-use device. The U.S. FDA's position on reprocessing by third parties continues to remain focused on proper reprocessing measures to primarily reduce the risk of patient infection during reuse and as well to ensure substantial equivalence to the predicate device with regards to functionality. In the U.S., the FDA's guidance documents for reprocessing draft all the way back to 2000 and continue to provide relevant guidance and information for the reprocessing of single devices. Additionally, the Medical Device User Fee and Modernization Act of 2002, commonly referred to as MEDUFMA, provides additional requirements, specifically in Section 302 for reprocessing of single-use medical devices. Again, these documents remain in place in the U.S. as relevant references for the reprocessing and labeling of devices. Looking at the MDR, prior to 2017, the 2017 revision of the MDR, the European Union did not have policies in place governing the reprocessing of single-use devices. With the latest publication, the MDR now has defined a regulatory pathway for the manufacturing and reprocessing of single devices. This regulation now allows a reprocessor to align their practice with a regulatory document to demonstrate their products are as safe and as effective as the original device, and thus meeting the same regulatory requirements as original equipment manufacturer. The constraints of the regulation, however, do require that the allowance for reprocessing be based on national law and not be overridden by the EU's acceptance of the MDR. Additionally, the regulation requires that the reprocessor, A, be considered as the manufacturer of the reprocessed device, B, assume the obligations incumbent on the manufacturers under the MDR, ensure an equivalent level of safety and performance to that of the corresponding initial single-use device, and lastly, the reprocessor must comply with aspects of the MDR Article 17 single-use devices and their The MDR outlines the requirements for single-use devices reprocessing in Article 17 of the document. The Article 17 is made up of 10 subsections, and in the articles define a minimum requirements for reprocessing in the EU. Specific requirements are as expected, such that the reprocessing party shall be considered the manufacturer of the device and shall assume the obligations incumbent on the manufacturer laid down by the regulation, basically stating that they assume all legalities and responsibilities of the device moving forward. Other such clauses, such as 3, allow exclusions or excursions from certain rulings, providing that the reprocessor is performing in accordance with common specifications, detailing requirements concerning such matters as risk management, the validation of processes and procedures, product release and performance testing, a solid quality management system, reporting incidents involving devices that have been reprocessed, and lastly, the traceability of the reprocessed device. Additionally, as expected, clauses relating to label and requiring that if the device is a single-use device, that it has been reprocessed, the label indicate the fact that it has been reprocessed, including the number of reprocessing cycles already performed and capturing any limitations to the number of reprocessing cycles. The MDR also imposes timelines related to cutoff dates, such as in Clause 6, which state that only single-use devices that have been placed on the market in accordance with the regulation or prior to May 26 of 2020 be reprocessed. Overall, the content in Article 17 and the MDR in general for single-use device reprocessing can be considered a significant step in the right direction for reprocessors, again providing regulation to ensure safe and effective reprocessing devices make it to the market. Again, there's 10 clauses that are a little bit too much to go into detail in today's webinar, so I encourage you to, again, look at the MDR or download it as ALPA stated and take a look for yourself and see what applies. So that concludes today's webinar on cleanliness testing for medical devices. I now turn the presentation back over to Chris for the closing and Q&A session.
All right, thank you, Paul, for a great presentation. Also, thank you to Alpha and Alexa. And now, before we begin with today's Q&A, please direct your attention to our webinar survey available on the right-hand side of the presentation window. If you close the survey, you can reopen the widget by clicking on the icon along the bottom of your screen. And thank you in advance for filling out the feedback form. Your participation in this survey allows us to better serve you. We're now going to move on to the question and answer portion of our event. As a reminder, to participate in our Q&A session, just type your question into the text box located to the right of the presentation window, or click on the Q&A icon at the bottom of your screen. And please note that we'll try to get to all the questions that are asked today, but if we're not able to get to your individual question today, we'll be sure to share them with our speakers, who will reply to you offline after the program is over. So let's move on to our first question here. And it looks like our first question is going to be for Alpa. Alpa, can you comment further on the proposal to modify the Spalding classification that has been so universally accepted for so long? Interested to hear the proposed changes.
So this is a good question, but the Spalding classification is will be only applied to the endoscopes, and we're not changing the Spalding classification. We're just adding endoscopes to that portion where it will be in the realm of critical devices, and move from semi-critical devices. So it will be the same, but there's just discussion on where do these belong now. Should we keep them in semi-critical because they do go through, they are gone through mucosal membranes, but they do breach skin when they're using catheters and stuff through it. So, or biopsy forceps, and they're pulling it out. It is still coming in contact with blood and tissue. So that's the only thing that I was talking about.
All right, thank you, Alpha. Our next question says, will the new TIR 12 consider European requirements as well, specifically Germany?
The new TIR 12 is very preliminary at this point. It did go from 7 to 3 proposals. However, I think it will, because the co-chairs that are part of this document are also in ISO, and I will bring this up in our next meeting to see if it will be considered.
All right, thank you, Alpha. Looks like we have another question for you here. It says it appeared that ineffective drying of the lumens was impending EO sterilization of the scope. Has there been work done to ensure adequate drying prior to sterilization?
This is a good question. During the stakeholders meeting just a few weeks ago, this was one of the topics that nobody had an answer to. And it was a difficult topic because these are high-level disinfected scopes, and how do they get dried? They're dried in a cabinet. And usually they do have forced air that's put through and flushed with 70% IPA to facilitate the drying. However, there is no specific criteria on how these scopes should be dried. One of the things that was proposed by some of the committee members that were there is that we should have adapters that are connected to the lumens and put them in a filtered hood where there is flow going on them through all the time while they are sitting in the cabinets. So the lumens are bone dry at that point. However, there is no decisions based on that, and hopefully in October we will discuss about the drying.
Okay, thank you, Alpha. Next question here: is there a specific standard that we can use to refer to when deciding, test soils for cleaning, validation, or reusable medical devices?
Yes, the ASTM F3208 was just published in March, and that goes through all the test soils that are used for reusable medical devices. It also categorizes what type of devices you can use. What type of test soils you can use for what type of devices. So it is a very, very good document that can give you information on what type of test soil should be used for the validation. ISO 1583-5 also has a bank of test soils in there, but they're not that helpful when it comes to selection or to figure out what is a perfect test soil for my device. So that's why this ASTM document was placed. It took us 5 years to get there, but we finally did, and it's a very good document to reference when deciding for test soils.
All right, thank you, Alpha. Another question here: were the AAMI TIR 30 benchmarks for cleaning, or were they updated? The AAMI TIR 30 benchmarks, were they updated for cleaning?
No, they have not been updated yet. I don't know if they will be updated. There is discussion on papers or even literature that can help with defining what the benchmark acceptance criteria would be, but at this point, it hasn't been updated yet.
Okay, thank you, and we'll move on to our next one. If we've already have a documented cleaning validation for our orthopedic device, would we be required to comply with this new ISO 19227 document?
Thanks, Chris. This is Alexa taking over that question. So yeah, that new ISO 19227 document, I would say if you already have a documented cleaning validation on file, that I would go ahead and read through the standard anyway just to be up to date on, what ISO is requiring. And I would actually hope that the validation that manufacturer would have on file wouldn't actually be too different than what the ISO is requiring. I would think that it would have to be, risk-based, where you evaluate all the different potential residuals and contaminants that are there, and then the ISO just has recommended test options. So I would go ahead and guess that it probably, the validation wouldn't be too different from what the ISO is suggesting. And if not, I actually might recommend some sort of gap analysis, or if the ISO is recommending something that you haven't done, that you evaluate maybe why you wouldn't need to and have that documented. So I would definitely pay attention to the new ISO document as it comes out and evaluate your cleaning validation just to make sure that it would stand up to any audits or questions that come your way.
All right, thank you, Alexa. Our next question here looks like, how would you establish specifications or acceptance criteria for manufacturing residues or containments, contaminants?
That is an excellent question, establishing acceptance criteria for manufacturing residuals and contaminants. And I could probably spend a whole nother hour talking about the different options for this. But I will say in general, we kind of used to use all of the tests together, kind of work together. So you quantified residuals with different analytical methods, and you tested your microbiological contamination and had cyto in there. And if you had, passing levels from the other tests, that would help you. And we used to consult on, using comparisons like your clean to dirty and showing a reduction in residue, and that's probably been the way that we've recommended to set acceptance criteria in the past. And to my knowledge, the new ISO document, it gives some guidelines in there for acceptance criteria, and that had a lot of debate. As I mentioned, the comments were coming back. The acceptance criteria that they had in there, I think, received the most feedback and comments regarding it because from my perspective, manufacturers want to be able to set their own acceptance criteria and not be locked into anything. And it's been difficult to set because so many devices, there's so many different types of devices out there and materials and processing, and it really comes down to, for me, what your device is and the intended patient contact and duration. And so here at Nelson Labs, we've been recommending people go more the ISO 10993-17 route, where you try to specifically identify and quantify the residuals that are left behind in your device and then do a risk assessment if that were to come in contact with the patient for that specific route of administration and duration. Would that be considered safe or not? And so I might recommend utilizing, toxicology, utilizing the services of a toxicologist to go ahead and evaluate those for patient safety. I think that's becoming more of the norm to really set standard acceptance criteria for these types of residuals. So hope that helps.
All right, thank you, Alexa. We have another question here. Is there a standard that outlines the determination of the number of units, for example, endoscopes, that constitutes a statistically relevant sample number for cleaning validation?
Thank you, Chris. This is Alpa, and I'll take this question. Unfortunately, there is no standard that says statistically relevant sample number is such and such. However, TRAD 12 and TRAD 30 do specify that a minimum of 3 test articles, 1 positive control and 1 negative control, should be tested. And that's what has been tested for almost all the cleaning validations that are performed. As for endoscopes, all the endoscopes are also the same as reusable, so they also require at least 3 test articles that are processed through the cleaning process, a positive control and a negative control. However, for duodenoscopes, you have to have 9 replicates, and the reasoning for the FDA to ask for more is because those were the scopes that caused the infections where the deaths occurred in 2015.
All right, thank you, Alpa. Looks like we have one final question here. Was there any discussion regarding the timeline to when the shift from HLD to sterilization will occur for endoscopes?
At this time, there is no timeline set out. We're looking at least when we were at the stakeholders meeting, the presenters did propose that in 5 years we should be able to transition to sterilization. We have to give industry to get into the validations and to understand what kind of sterilization modalities are available for endoscopes. And they did say they did mandate the FDA that it should be within 5 years, and that should be our time cutoff. But of course, this will take a long time, and it's not going to be a quick transition.
All right. Well, thank you, Alpa, and thank you, Alexa, and also Paul for a great Q&A and presentation. That looks like all the time that we do have for questions for today. I'd like to inform our audience that within the next 24 hours, you'll receive a personalized follow-up email with details and a link to today's presentation on demand. Please feel free to invite your colleagues and peers who may not have been available to listen to today's event. And also, as a reminder to our audience, if we didn't get to your question today, someone will be getting back to you via email after the program is over. And this webinar is copyright 2017 by UBM. The presentation materials are owned by or copyright by Nelson Laboratories, and the individual speakers are solely responsible for their content and opinions. I'm Chris Keach. Once again, we'd like to thank you for joining us for this week's course, and we hope you have a great day.